Welcome to our webinar series for professionals.
So wherever you are in the world, hello and welcome to this event that we have. We have a stunning sign-up rate. So it's really nice to have everyone involved. So I'm just going to do my introduction. So if you don't mind not sharing your screen now, so because then I'm on full screen, and then as soon as I hand over the screen to you, then you can start sharing the screen. From the Philippines, wow, and Germany, Norway, Turkey. I love this language thing. You know, I love the world. I was just in the US last week and meeting all sorts of people, and it's just so nice. Anyway, it's just past 4 o'clock Copenhagen time. I'm Anne-Catherine, co-founder of Nordic Laboratories and DNA Life. As you know, we are a group of companies that are dedicated to healthcare or change healthcare through functional medicine and personalized medicine. And remember, you have your practitioner portal to go to, VMS, where you, of course, can find the tests that we are talking about today. Reach out to us if you need help. We are not far away, and we are eager to help as much as possible. So today we have a special guest with us, and now I'm just losing my notes here. So today we have Dr. David Zava with us on board. He's the star of the show today. Last time I met David was in Austin, Texas, actually at an IFM event. He was having breakfast with his wife, I remember. So anyway, Dr. Zava is a biochemist, a researcher, and he's the president of ZRT Laboratory. And he founded ZRT nearly 25 years ago. And he has a very humane edge to the way of running his business, because he wants to provide practitioners and patients with a deeper understanding of the role of hormones. But he wants to channel his knowledge into enabling people to actively be involved in their own health. And that leads me to remember that, who owns your health? Is it your doctor? Or is it your parents? Or is it really you? And I have also in some of the other webinars used the N=1, and we are responsible for our own life experiment, really. So, but having said this, the topic of today is focus on thyroid and thyroid hormones and nutrients related to these biochemical processes in the body. So remember, we are recording, and we will share the recording. We will also share the slides as soon as we can. So sometime next week it will come out to you all. And the recordings will also be in VMS if you log on to VMS. So for now, I will pass on the stage to Dr. Zava. Remember, you can, all those nice people saying hello from all the different countries in the world, keep writing to each other. Also ask questions, because at the end, when Dr. Zava has done his presentation, there will be time for Q&A. So write your questions in the notes on the chat box. I will take note of it. And at the end, I will ask the questions and take it from there. So I will mute myself, Dr. Zava, and then I will pass on the stage to you.
Okay, can everyone see the screen now?
You can see you, but not your screen. So you need to share the screen.
You see that now?
Yes, we can see it now. Absolutely perfect.
Okay, is that the full screen or is that the partial screen?
It's the full screen. It looks
okay,
good.
Great. All right, well, welcome everyone around the world. I'm in Portland, Oregon, sitting at my desk where I do most of my work, and I had to clear everything around me because I have lots and lots of papers all the time reading. I'm a biochemist. Really, my expertise started with breast cancer, but because I got into breast cancer, there were many different things that I was interested in, and thyroid and heavy metals was certainly one of these things. And you'll see at the end of my slide that sort of pull that together with regard to my interest in breast cancer. But thyroid is a difficult subject, as most of you probably know. You know, you measure thyroid hormone in the bloodstream, and you probably often see patients who are have perfectly normal thyroid levels, and yet they suffer from a lot of thyroid deficiency symptoms, and that is related more to the cells, the tissue levels of in your body that are probably not responding properly to the thyroid hormone that your thyroid gland is making. So we're going to sort of get into the thick of thyroid with regard to essential elements, selenium in particular, and iodine, and their relationship to exposure to heavy metals that will affect not only thyroid hormone synthesis, but the metabolism of the thyroid hormone, as well as the function of the thyroid hormone. Let me figure out how to get this to. Okay. So the topic of discussion today will be iodine and thyroid hormone synthesis, selenium and thyroid hormone synthesis and metabolism. Selenium is essential for thyroid. Selenium, as it relates to thyroid deiodonases and antioxidants, and the impact of environmental elemental toxins on arsenic and mercury. So let's look first at just iodine deficiency and iodine deficiency disorders and how that relates to the thyroid hormone. So if you don't have enough as you're developing as a fetus, and you don't have enough iodine, there can be problems for the fetus. It can cause a miscarriage, stillbirths, congenital anomalies such as cretinism, and that's excuse me, got a frog in my throat. Congenital anomalies such as cretinism, brain damage, mental retardation, increased perinatal morbidity and mortality. So there's a lot of things that can happen if you don't have adequate levels of iodine in your in the mother during growth of the fetus. Once the baby is born, iodine deficiency with regard to the neonate can cause endemic cretinism, neonatal goiter, neonatal hypothyroidism, and endemic mental retardation. So you definitely need iodine. You need iodine because that's what you use to make thyroid hormone. Iodine deficiency in the child and adolescent can cause goiter, can cause subclinical hypothyroidism. It's kind of a lowish T4 and higher TSH. School performance and IQs can be impaired. There's retarded growth, hearing, speech, the ear. You know, people may not realize it, but hearing impairments are in part can be caused by a deficiency in thyroid, which could be caused by a deficiency in iodine. So by the time you get to be an adult, I don't see people like this walking around, on the streets, but in certain areas in Indonesia where they eat certain foods that suppress the incorporation of iodine, you can get goiter. The certain areas in the United States and Switzerland, other countries had iodine deficiency and goiters until about the 80s and 90s when they started supplementing the salt with iodine, so there wasn't such a problem with iodine deficiency. But goiter, apathy, overt hypothyroidism, which is low T4 and high TSH, and it could be moderate, borderline low T4 with moderate high TSH, suboptimal brain function, sluggish thinking, low capacity for initiative and decision-making, spontaneous hyperthyroidism. The reason for that is because if you got a thyroid that's this big, it's because you haven't had any iodine and the thyroid gland is being stimulated to grow and get larger so it can make more thyroid hormone because it's really pushing hard. So there's a hypertrophy of the thyroid gland. So you can actually have a spontaneous hyperthyroidism. And even iodine itself, believe it or not, when you have too much iodine, it can cause hyperthyroidism also. So if you had a goiter like this and you gave the person iodine, they may be synthesizing huge amounts of thyroid hormone at that point, become hyperthyroid. So let's move from iodine to selenium. Selenium, I think, if you don't leave with anything else, it's important that you realize that selenium is essential for thyroid hormone synthesis. Thyroid hormone synthesis would not occur without selenium. And there's several places where selenium plays an important role. The enzyme that takes iodine and puts it on the tyrosine residue of thyroglobulin is a glutathione peroxidase and thioredoxin. They both are important for protecting the thyroid peroxidase enzyme. So it requires peroxide, and it takes iodine and ionizes it and puts it on a tyrosine residue. So you get four iodines, and you have T4. You have if it only puts 3 on there, you could have T3. So the selenium-dependent glutathione peroxidase and thioredoxin reductase protect the thyroid from potential damage caused by the peroxide. Peroxide is extremely toxic if it's left alone in the body in a high concentration. And the thyroid makes a lot of peroxide in order to carry out the reaction of putting iodine on the thyroglobulin molecule to make thyroid hormone. So that's the synthesis process. It's a very hot process. It's a very energetic process, and you could, if you didn't have selenium and the selenium-dependent enzymes present there, it could damage the thyroid. Also, for extrathyroidal tissues, selenium is present in the catalytic site of all 3 thyroid deiodonases. Thyroid is released, so mostly T4 and some T3 is released from the thyroid gland. The T4 has got to be converted into T3 because that's the active hormone. I mean, I think of like estradiol and estrone. Estradiol is the active molecule, estrone is the inactive molecule. Cortisol and cortisone. Cortisol is the active molecule. Cortisone is the inactive molecule. T4 is the inactive molecule. T3 is the active molecule. So all systems work that way. They convert back and forth. So you have to be able to convert T4 into T3. And a lot of that is occurring within the target tissues themselves. That conversion requires an enzyme that a deiodinase, and all 3 of the deiodinases that convert T4 into T3 are selenium dependent. If you don't have enough selenium, that won't occur. So selenium dependent deiodinases are essential for activation of T4 to T3 by both D1 and D2. D2 is really probably the most important in terms of the total amount of T3 that you're making in your body. And also, under conditions of excessive stress, you can have your body then makes more of the D3, which converts T4 into reverse T3. You all have heard of excessive amounts of reverse T3. Well, that just shuts the system down. It's interesting, if you look at papers like this, you realize that D3 is located really on the outer surface or just on the inner surface of the plasma membrane of cells. And so when T4 comes along, D3 will convert it into reverse T3. And so reverse T3 has really no affinity for the thyroid receptor, and it really is not getting into the cell, and it's not increasing the pool as you see the little basketball on the inside of the cell. It's just showing the amount of T3 that's present, as opposed to the bottom diagram. You're seeing T4 through D2 being converted into T3, or T3 itself increasing the intracellular pool of T3. That T3 then binds to thyroid receptors, and that activates the genome. That's where the real action is occurring. We don't necessarily see that when we measure thyroid hormones out in the bloodstream, but we get a pretty good idea of whether or not you're making adequate amounts of it. So D1 and D2 are, as said, located in the plasma membrane, whereas D2 is in the endoplasmic reticulum, so it's inside the cell. D2 generates T3 in the cytosol, so it's ready access to the nucleus and thyroid receptors. T3 half-life in plasma is minutes. So when you take a combination T4, T3, T3 doesn't hang around very long. It's very rapidly destroyed, and that may depend on the amount of thyroid binding globulin, which binds very tightly to T3 and to T4 and keeps it in the active in the bloodstream, but it's not very bioavailable. A very small percentage of T3 and T4 are present. So when you take estrogen and you take estrogen in excess, your liver makes more thyroid binding globulin, so you have less bioavailable thyroid hormone. But we're focusing now on thyroid deiodonases. So D2 half-life is about 40 minutes. So it's decreased by the interaction with T4. It's called ubiquitination. It's interesting because I worked a lot in biochemistry area where the enzyme thyroid deiodinase, D2, once it binds to T4, it actually inhibits the enzyme itself after it begins to convert. So the enzyme doesn't hang around for a long period of time. So it has to be resynthesized within the cell. D2 produced T3 is a significant physiologic determinant of energy expenditure in humans. A larger metabolic role of D2 in humans may be anticipated because skeletal muscle is a predominant site of thermogenesis and insulin-induced glucose disposal in human adults. So it's, your muscles, particularly muscles in your legs, are important for thermogenesis, production of heat in the body, because as your thyroid works, it creates heat. Humans also express D2, a lot of it in the myocardium, so the heart, important, which could make the myocardium more sensitive to changes in circulating T4. There's a lot of genes that are regulated by T4 to T3 conversion in the heart that makes the heart important. Selenium is important for, as I said, glutathione peroxidase, and that also helps. This is a lot of these are complicated slides. I'm just trying to simplify what all this means. But what this means is that selenium is important for production of glutathione. Glutathione is the predominant antioxidant in the body. And so you need selenium for keeping your glutathione levels at a higher level. So questions that I'm sure just burning in your mind. Since selenium is vitally important for thyroid hormone synthesis and metabolism, the conversion of T4 to T3, is selenium deficiency or excess a health problem? Is it? Certainly, there's lots of literature on that, and then we'll talk about that in a minute. If selenium deficiency or excess is a worldwide health issue, can it be measured in body fluids, urine or blood, and does this reflect dietary intake? Is thyroid dysfunction related to environmental toxins interfering with selenium availability in thyroid deiodonases? And we'll get into that and talk about heavy metals such as arsenic and mercury and its relationship to selenium bioavailability. In certain areas of the world, there are selenium deficiency diseases associated with where you live. So, for example, regions of China and other parts of the world have both severe selenium deficiencies and excesses and diseases associated with both. So here's a reference for you all to, look at, and you'll get access to to all of this information. But China's interesting. You can have on one side of a mountain, you can have a selenium deficiency, on another side of the mountain, you can have a selenium excess. So, but for the most part, selenium deficiency diseases, such as Kishan disease and Kashin-Beck diseases, are prevalent in those areas where there isn't adequate amounts of selenium. And then you can have areas where you have excessive amounts of selenium and you have selenosis. If some of you may have remembered when some polo horses from Italy came over to the United States and they were injected with selenium and they got the concentration wrong by 100-fold. All 24 of the and selenium is very good because it's very good for your muscles, but not at 100-fold higher concentration. All 24 of those horses within a day or two died because they were given excessive amounts of selenium, and selenium is not good for you beyond a certain point. So if you don't have any selenium, that's really bad, slow disease. But if you have way too much selenium, that can be very toxic too. So, China has some areas where there's selenosis, excessive amounts of selenium, and some areas where there's a deficiency of selenium. With regard to Japan, they've taken a look at dietary intake and urinary excretion of selenium in a study. Basically, what they found is that the daily urinary selenium excretion was the most discriminating indicator of short-term selenium intake relative to plasma or red blood cells. So it's a good way of determining if you're getting enough selenium. So your average diet, it reflected your selenium intake. But that's important. So you can, collect urine. And what they found is that urinary selenium may be a more convenient indicator of selenium status than plasma selenium. They both work. We do both. So we do selenium testing in urine. So it's just a matter of urinating, in a cup, dipping it into a filter card, and then drying it. It allows for easy transport of the specimen because it's dried. So there's no liquid present. It's easy to ship, and iodine and selenium are extremely stable. You can come back a year later, you can leave it out, come back a year later and still measure the levels. But this is just looking at selenium in human urine. It's a review. It was published a while back, but I don't really see any difference in what they're showing in the healthy controls here. And what we find, ZRT reference range mean was 55 micrograms per gram of creatinine, and that's basically what they found in the healthy controls. And so you can look at all different sorts of cancer patients, et cetera, all the way through here. When we have looked at cancer patients, they generally tend to, in certain areas of the world like China, have lower levels of selenium, but they also have higher levels of heavy metals and other things like that. And we'll talk about that, what keeps you from having adequate levels of selenium. So here's Selenium in Human Health and Disease, published in 2011. You really don't want if you're looking at blood levels of selenium, you don't want your selenium to get over about 250 nanograms per milliliter. And that's about what we see. Median levels are somewhere right where they should be in the United States. And other areas of the world, it can be much lower depending on the amount of exposure to other toxic elements. And again, we'll talk in a few minutes about how selenium forms complexes with both arsenic as well as mercury. So you go back historically and you look at, what scientists were thinking about selenium, and in the 1930s, they believed that selenium was highly toxic. Well, it is at high concentrations. So are a lot of other things. But by the 40s, they believed that selenium was a carcinogen. And by the 50s, they realized that selenium was an essential element. By the 60s, they realized it's an anti-carcinogen. And by the 90s, they realized selenium is essential for thyroid hormone synthesis and metabolism. So that's kind of the evolution of how people were thinking about selenium. So selenium is really important for thyroid hormones, and also as an important antioxidant in enzymes that are involved in removing toxic elements. So when you look at selenium in human health, you can see that diseases basically go down when you get to an adequate amount of selenium, but they could go back up again if you have excessive amounts of selenium. So again, I could spend all day. I could spend an entire hour on each one of these references that I'm showing you. I'm just trying to give you, the basis, and you can look at these references yourself. Hashimoto's disease. We know that Hashimoto's is closely associated with selenium deficiency, and that Hashimoto's is where you have thyroid peroxidase. The body is attacking itself, so it's attacking the thyroid and releasing thyroid hormones because the immune system has said, okay, well, I find that thyroid peroxidase, which is the enzyme in the thyroid gland that actually synthesizes thyroid hormone. So you can get, and as I showed you earlier, selenium is very important in the enzymes that are protecting the process of formation of thyroid hormone. So these are the antioxidant enzymes that are present in the thyroid gland that protect. So in Hashimoto's disease, selenium supplementation appears to potentiate the activity of selenoproteins, thereby decreasing local inflammation, because Hashimoto's is an inflammation, and people get flares if you have antibodies, and we measure those in the bloodstream because the immune system is making antibodies to the thyroid. So it attacks the thyroid and it causes damage to the thyroid and becomes fibrotic eventually, and then there's no more thyroid left. But during those storms, thyroid storms, you can have a large release of thyroid hormone into the bloodstream, and then it stops. So you can have a storm, you have an excess of thyroid hormone followed by deficiency. And that's kind of what is happening with Hashimoto's disease. So it's thought to be associated closely with the availability of selenium. It doesn't completely remove the problem of Hashimoto's disease, but it can lower the degree of inflammation in the thyroid storm. So let's move on to environmental toxins. What might interfere with iodine incorporation into thyroid hormones T4 and T3, and selenium incorporation into selenium-dependent deiodinases. So those are the ones that are converting T4 into T3 or reverse T3. Or just simply antioxidants, glutathione peroxidase, the thioredoxin reductase. These are selenium-dependent enzymes. They have selenium at the catalytic site of the enzyme, so that's where the action is happening. So without selenium, these enzymes, these antioxidant enzymes, are not going to be present, or going to be present in much lower concentration. So important to realize that selenium has a very strong affinity for arsenic and mercury. So here's where we're bringing in the toxic elements, because when we first started doing thyroid hormone testing, we looked at iodine, we looked at selenium, And then we began to look at heavy metals. And I've been sort of involved in that area and interest in that area for a very long time. So, we were able to develop the assays on either a dried urine sample or a dried blood spot sample. So, and that works extremely well. I don't have time to go into the details of that, but we can see the concentrations of the heavy metals as well as iodine and selenium in the same prep, in the same sample that were given, be it either dried urine or dried blood. So important to realize that arsenic and mercury bind with really high affinity to selenium. It is an ionic bond that you almost couldn't blast away with a nuclear bomb. It is an extremely tight affinity, and it just basically, once it occurs, it's not gonna release. So it's gonna take up the selenium, and you will end up excreting it. It's not going to be able to get into the thyroid, the deiodonases or the antioxidant enzymes. So when levels of these heavy metals are high or selenium is low, this results in lowered levels of selenium-dependent antioxidants such as glutathione peroxidase and thioredoxin reductase and thyroid deiodonases. So all of these things that are important that have selenium in them are going to be lowered. So when you look at a result and you see, okay, it's low normal selenium, but then you've got toxic elements that are high like arsenic or mercury, the bioavailable level of that element, such as selenium, is going to be much lower. You're just not going to have as much of it there because it's tied up mostly by the heavy metals. The heavy metals are, we all know, they're bad for you. So lower thyroid deiodonases, the D1, D2, D3, can result in lowered cellular conversion of T4 to T3 and hypothyroidism. So a lot of this is going on within the cell. So as I said, D2 is intracellular, it converts. All cells of the body have it. It's a lot of the thyroid hormone that you make and that you use is what's happening inside the target cells, which again is every cell of your body. And if you're burdened with heavy metals that are binding to these deiodonases or preventing you from having adequate levels of glutathione peroxidase and thioredoxin reductase, then you're going to have less of the thyroid hormones, and you're going to have fewer of the antioxidants. So lower antioxidant capacity can lead to higher levels of reactive oxygen species. And we know about reactive oxygen species, heavy metals causing increase in reactive oxygen species, increase in cardiovascular disease, and I'll get into that in just a little bit, increase in cancer, Hashimoto's as an example of what's happening in the thyroid gland itself. So it can lead to diseases such as cancer of the breast and cardiovascular disease. Books have been put out. I like this one by Isabella Wentz on thyroid diet, on selenium deficiency and the thyroid. Selenium status, thyroid volume and multiple formation in the area of mild iodine deficiency. So in this paper, basically they showed that serum selenium concentration was found to be significantly negatively associated with thyroid volume, and a low selenium status significantly increased the risk for thyroid enlargement. Furthermore, low serum selenium status has a tendency to increase the risk for development of multiple nodules. So important to realize that selenium and iodine work in concert with one another, and iodine can be a little more toxic if selenium isn't present. They work in harmony with one another. Selenium allows iodine to do all of its beneficial things And selenium prevents it from having any kind of a toxic effect on the body. So that's kind of the bottom line to that. So, they've done studies, looked at the toxic effects of iodine. I don't have it in here, but we did a study in China, which I'm co-author on, and I should have had it on here, but where we looked at iodine levels in relationship to thyroid issues such as goiter. Too much iodine can also, as well as deficient levels of iodine, can cause goiter. You can have a Wolff-Chaikoff effect that results in excessive levels of TSH. I've seen a lot of very high TSH levels in some people that go on excessive levels of iodine. I'm one of those people, and I test myself about once a week. And you've heard I've been doing this for 25 years, so a lot of tests that I've done on myself. And I know when we were developing this, we were taking higher levels of iodine, and I'm one of those people that tend to make a lot of TSH. So I have a relatively, I take thyroid hormone because it runs in my family. And my TSH, if I don't take thyroid hormone, is 7 to 8. If I take it, I can get it down to 1.5 to 2. If I take excessive amounts of iodine, my TSH can go up to 20 and go way high. So that doesn't happen in everyone, but it does happen in a subset of people who are, I guess, iodine deficient. My mother always said, "Oh yeah, I'm iodine sensitive," probably related to what she was talking about, but I don't think she had the details of what we're talking about here. So selenium is important for helping to prevent the toxic effects of excess of iodine. As I said, selenium is important because it increases the enzymes that will keep iodine from making toxic metabolites and causing goiter. It doesn't happen in everyone. It's not like it's 100%, but it does happen in a subset of people. And you can look up my name and look up, that study that we did, but it can lead to abnormalities and nodules in the thyroid gland. And this is just a continuation of that study. So let's move on a little bit to selenium interaction with heavy metals, arsenic and mercury. What are some of the health consequences when selenium is low and heavy metals are elevated? Because we see this, I've seen this in certain areas. We measure these things because it's easy to ship a dried sample, from Timbuktu to Portland, Oregon, all over the world. And so we have pretty good information on people who live in areas where there's very heavy contamination of the water systems or whatever, the soil. If it's in the water, it's in the soil. You're watering plants, they're going to take up these elements. For example, in growing rice, if you grow rice in an area and you irrigate it with water that contains arsenic or any other heavy metal, it's going to take it out. And the rice is going to contain the heavy metal. So we know that in certain areas of the world, people are cautioned that the rice may have high levels of arsenic. And, you've seen this published in many different journals. So there isn't adequate levels of necessarily adequate levels of selenium when the arsenic levels are high. Arsenic is a problem in well water and food. For example, rice irrigated with high arsenic water. So you can get quite high levels of arsenic in the food, such as rice. And again, lots of in the last 10 years, articles have been published on higher arsenic levels in rice that's coming from areas where arsenic is relatively high. In China, 2 forms of endemic chronic arsenic poisoning are common in drinking water and coal smoke pollution, inhalation of smoke. You have to realize that when you dig deep into the earth, you destroy Mother Earth because you're mining coal. Coal contains a lot of heavy metals. It contains mercury, it contains lead, it contains not so much lead, but arsenic for sure. And that this then gets into the atmosphere. Rain brings it down. It gets contaminated in the soil, and then the plants take it up and you eat it. And as I said, there are areas of the world, and for example, in China, where you have high levels of selenium and you have low levels of selenium. So in a study we did, we certainly found that in the breast cancer patients that we were looking at, the selenium levels were exceptionally low, and heavy metals were high. So when arsenic is excessive, it's been published that it's associated with increased incidence of cardiovascular disease. More than 100 million persons are exposed to arsenic levels in drinking water above the WHO standard of 10 micrograms per liter. Urinary arsenic measurements integrate all sources of exposure at the individual level, including water and food, and are an excellent biomarker of internal dose. So you find out if your arsenic is high. We see people, we certainly see people with high arsenic levels. You have to be careful, because if you're looking at arsenic and someone's eating shellfish, it could be arsenobutane that's high. So they should avoid eating any shellfish the night before they do their test. That clears very fast, but arsenobutane, which comes from shellfish, and is not toxic. It is a derivative of arsenic. Is not problematic. If I eat shellfish and I do my arsenic levels, and my arsenic levels tend to be higher. Exposure to low to moderate levels of inorganic arsenic, that's the bad one, as measured in urine, was in this study prospectively associated with increased fatal and non-fatal cardiovascular disease. And this was done in the United States. You can take a look at this paper. So arsenic's not good. Treating chronic arsenic toxicity with high selenium lentil diets. So there are certain areas of the United States and the world where there is quite a bit of selenium in the soil. For example, the plains in Canada have a lot of selenium, and they grow lentils, and they have really high levels of selenium in them. So it's good. Selenium, the best way to get selenium into your body is through the food you eat. You can increase your selenium, but the best way to get it incorporated into all the enzymes we're talking about is through food. And so yeast, as well as lentils and foods of that nature, are really good for increasing your selenium levels. So treating in this study, lentils grown in selenium-rich soils increase the antioxidant capacity. So they increase the level of glutathione and increase T4 to T3 conversion, selenium-dependent D2. So these are all the things we've talked about. So by increasing, selenium levels in the body, also for immunocompetence, with viral diseases threatening us every day, from COVID to, monkeypox to whatever is the next one in line. Certainly adequate levels of selenium are necessary for as an antioxidant, and also for your immune system. Toxic effects of arsenic in water reverse the selenium in food, increase arsenic excretion in urine and feces. So selenium, you're going to get more excretion in both in urine and feces when you have adequate levels of selenium, and I'll show you what's happening there. But let's first talk about mercury. Mercury is going up. You burn coal for fuel. And certain countries of the world are burning a lot of coal for fuel. We're trying to get away from that, but it's a health problem because all of that smoke that goes up into the atmosphere has got a lot of mercury in it. Coal has a huge amount of mercury in it. And we also think about mercury in fish. Most fish from the sea, the selenium mercury ratio is about 1. So seafood, where you have the selenium binding to the mercury and basically inactivating it, so seafood is not going to be as toxic as a fish that you might eat from a Great Lake, which has a high level of a toxic element like mercury and a low level of selenium. So seafood in general has a ratio of selenium mercury of about 1. And that reduces the mercury toxicity. So eat your seafood, no problem. It's less toxic. Seafood is safer to eat due to the inactivation of mercury by selenium combined with the beneficial actions of omega-3 fats. We all know that. So I'm gonna, you'll have, like I said, you'll have access to these references, but I'm gonna skip over these for time. So here again, selenium is, and the incorporation of selenium into selenoproteins is very important for thyroid hormone action in the deiodinases. I thought this was really interesting because this was a study done with looking at dental amalgams versus composite. And if you focus on the bottom diagram, so the composite, this is mercury levels in the y-axis here, and this is males and females. So left side males, right side females. You can see that with composites, the mercury levels are about the same. They're steady, as opposed to these were 8 to 10-year-old kids, male on left, female on right, that had two or more than 2 amalgams. So here's where the mercury levels were in their urine in people who had amalgam. So they took their urine. So you can see that if you have a composite, which is basically just plastic, versus a mercury amalgam, the mercury amalgams were resulting in higher mercury levels in these kids. It's definitely statistically significant. But as these boys grew older, they excreted less mercury as opposed to the girls. Girls excrete more mercury. So they can detox it better. So, and this is really, so if they're 10 years old, they're now, 17 at the time the study ended. So we can say that prolonged exposure to elemental mercury may cause impairment of the developing of the central nervous system. We know that happens, lots of literature on that, along with attendant personality, motor function, and behavior problems. And I find this interesting because males are more susceptible to developing ADHD. We're involved in a study right now at the medical school looking at different types of things, but the relationship of ADHD to nutritional supplements and things of that nature. But males retain more. In other words, they excrete less mercury and more likely. And we know males are more likely, 5 times more likely to develop autism. So what's happening? Is that playing a role? It probably does play a role. We know it's not the only thing that's involved in autism, but certainly for these kids, you don't develop autism when you're 10 years old. You've already developed autism at a much earlier age. So, but what about the parents that are consuming the different kinds of things? So ADHD, they did another study in Toronto University, where they looked at children with amalgam fillings who had had them for 2 or more years. And what they found was the same in the other study, and that is that kids with amalgam fillings had much higher mercury levels that they were excreting. They had higher serum levels of an enzyme, MDA, I forget the name of the compound, but it's an oxidation product of fats. So they had lower levels of glutathione and they had lower levels of GABA. So we think about GABA, and we certainly do urinary neurotransmitters. And so these kids had lower levels of this neurotransmitter that has a calming effect on the brain. They had less antioxidant capacity and they had lower levels of GABA. What they found in these kids was there's no difference in their IQ, that they were more withdrawn, more anxious and depressed, more social problems, greater attention problems, more likely to have delinquent rule-breaking problems. We're talking about ADHD here, so certainly we don't want to have kids with amalgams, because that may lead to, in some kids who can't detoxify it properly, maybe they have a different diet, it creates more problems. So here's an interesting study where they've looked at this and found that the kids that have more mercury in their urine. So, studying mercury levels is, I think, is important. It's interesting. Iodine reacts with mercury, increasing its solubility. Now, when they looked at the effect of iodine on mercury concentrations in dental unit wastewater, that's where they first discovered this. And that they were using iodine, so Lugol's or iodine to clean off the surfaces in dental offices. And they realized that when they used iodine, that because they were monitoring the amount of mercury in the sewer, and they realized that when they used iodine to clean the surfaces, to detoxify the surfaces, that they had more mercury in, because what iodine was doing was it was solubilizing the mercury. So iodine could also, solubilize mercury in your mouth or solubilize mercury in your body, but it also helps you get rid of it. It was interesting to me because I came upon a reference on mercury removal from Department of Energy solid mixed waste using the GEMEP technology. So it's basically using iodine to help dissolve mercury where there's huge contaminations. And as I read this, I realized, oh my gosh, the Department of Energy cites sediment from the East Fork of Poplar Creek, Oak Ridge. I was born in Oak Ridge, Tennessee. Oak Ridge, Tennessee was a site for nuclear. We did a lot of nuclear stuff. We made the isotopes for the bombs, and they would bring in box loads. I mean, you can imagine a train, box loads of mercury, because mercury was used in the processing of uranium, to generate plutonium. I don't know exactly, but it's probably in the centrifuges where they'd centrifuge the stuff through, because they had a lot of centrifuges there. And mercury, I remember my dad and I would go fishing close by, and we would, I'd say, well, let's go fish over there. We can't fish over there because they don't allow us to fish there. So he worked there. And nobody ever talked about any of this stuff. Nobody ever said anything. But they brought box loads of this stuff in. And so this East Fork, Poplar Creek was the same creek that flowed from these plants, these places where they were doing all of this work, through the city of Oak Ridge. And it went right by the high school. So, you know, those are creek I played in as a child. You know, am I mercury toxic? I think yes, probably. Yeah, I know I am. But it was, you know, it was a part of what was in the city. And I can tell you all over the world where they're doing this kind of stuff there, you know, anytime, any place you're burning large amounts of coal for energy, you got a lot of mercury. But we just, you know, in Oak Ridge, we brought in boxcar loads. I mean, literally boxcar loads of mercury. And I know there was a particular time when the mercury was spilling out of the plants and into the environment close by. So I was just a kid. That was a long time ago. You know, I'm 74 years old now. Can you imagine? I mean, that was a long time ago. So, but it's a problem everywhere. There's a lot of mercury out there. And, but I sort of stumbled on this because of the issue with iodine and iodine dissolving mercury. That's actually a good thing, but because it helps you eliminate mercury from your body. So let's go back to selenium deficiency or arsenic excess can lead to cardiovascular disease. So we talked a little bit about that. Heavy metals decrease selenium bioavailability to selenium-dependent antioxidants such as glutathione peroxidase and thioredoxin reductase and thyroid deiodonases. So that's a theme that keeps going through this. So how does selenium help prevent some of the toxic effects of both mercury as well as arsenic? Well, selenium binds with extremely high affinity. As I said, it's an ionic bond that's very difficult to break, which then bind to glutathione. Now, you all know, you're all scientists here, I'm sure, or had scientific training, to realize that glutathione is one of the most important antioxidants in the body. It's very important that you keep your glutathione levels normal. But if you have high levels of arsenic and mercury and really inadequate levels of selenium, you've got a problem in every single cell of your body. So how does that look? So if you look at this is the complex of arsenic and mercury with selenium. So here's selenium in the middle here. So selenium binds to arsenic. Arsenic binds the sulfur groups of glutathione. So this is perfect symmetrical interaction of glutathione. So now you're taking up your glutathione. So you burned up 2 molecules of glutathione. It's binding to arsenic, which is binding selenium, which is binding to methylmercury. So goes your glutathione. So goes your selenium. So this gets excreted from the body. But this is one of the complexes that forms in the body. So if you have too much arsenic or you have too much methylmercury in your body, it's going to bind up selenium. It's also going to burn up your glutathione. So you need to get rid of these heavy metals as much as you can. So what about the association between exposure to low to moderate arsenic levels and cardiovascular disease? We keep going back to that. High arsenic is going to result in low selenium. If selenium is borderline, so if you do your selenium test and you find that the level is borderline and your heavy metals are high, that's not good. You need to be able to try to remove the heavy metals, and one way of doing it is to increase your selenium. Also, iodine. Selenium-dependent protein thioredoxin reductase. So this is an enzyme it's really important for helping to increase the levels of antioxidant. It is an antioxidant itself, but also reduces CoQ10 or ubiquinone to ubiquinol. People don't realize that when you take CoQ10, you think, okay, I'm gonna benefit my mitochondria. If you don't have adequate levels of selenium, it's not going to work as well. because you have to convert ubiquinone to ubiquinol. The active molecule is ubiquinol, and that's cardioprotective. So, and here's a reference showing that the cardioprotective effect of coenzyme Q10 are most likely explained by its antioxidant effect, which requires continuous reduction of ubiquinone and regeneration to the active ubiquinol form. So in the mitochondria, this is going on continuously, and you have to have the conversion of ubiquinone to ubiquinol, and it's being burned up very rapidly in the body. So selenium is really important, and as I said, you have a lot of the CoQ10 is taken up by the cardiovascular system. It's very important. So you've got to have adequate levels of selenium. Well, as I said earlier, I'm a breast cancer researcher, and certainly this is really kind of how I got into iodine as well as selenium and breast cancer. And one of the ways that selenium plays an important role, we know that lower levels of selenium are associated with increased breast cancer risk. As I said, when we first were developing in around 2005, 2006, we were developing the urine estrogen metabolite assays. We were also looking at the levels of estrogen metabolites in people who were just diagnosed with breast cancer, and we also looked at selenium and heavy metals. And what we found in the breast cancer patients, as opposed to the normal healthy controls, is that the breast cancer patients had much lower levels of selenium than the controls. So selenium is significantly associated with breast cancer risk. Interesting, when they looked at increased rates of chromosome breakage and BRCA1 carriers. So these are patients that have BRCA1. So you're really high risk for breast cancer if you have the BRCA1 gene. And what they found when they looked at their just looking at their lymphocytes from these patients, where they could identify the damage to DNA in vitro in a culture system, is they found that the selenium, oral sodium selenite would actually prevent the damage to DNA. So BRCA carriers are, as I said, are particularly sensitive to oxidative stress. So oxidative stress from heavy metals, from everything in the environment. So what they believe is that the mechanism by which selenium reduces the frequency. So we're looking at chromosome breaks in the BRCA1 carriers who tend to have more chromosome breaks. What they found is that the selenium reduces the frequency of double strand breaks in these BRCA1 patients. Again, they're just much more likely to have those. So you can look at this paper in detail. What they also found was that their inhibition of 7,12-dimethylbenzanthracene. This is the very first compound I worked with. So when we were looking at breast cancer in rodents, we knew that you could induce breast cancer in these rodents by giving them DMBA. And I got real interested in DMBA. And the reason I got into the endocrinology was because of the link between estradiol. If you look at the structure of estradiol and look at the structure of dimethylbenzanthracene, it's kind of an aha moment. Wow, they look a lot alike. And we know that estradiol will form what we call catechol estrogens, and dimethylbenzanthracene. Part of my dissertation was to look and show that, in fact, actually there were this portion of the molecule was oxidized. Catechol estrogens will form here. So the 3,4 catechol is the bad catechol estrogen. And again, I'm more than glad to come back and talk about that. But we know that the formation of the catechol-estrogens is associated with increased risk of breast cancer. And in these studies on rats, what they found looking at dietary selenium is that the number of chromosomal breaks was decreased in proportion to the amount of selenium that was given to the rat. So you can see that there's this, and also a decrease in tumor incidence in these animals. So there is definitely a link to chromosomal damage if you don't have adequate levels of selenium. This is what we saw when we looked at the population of women who had breast cancer. I was shocked when I saw how low their selenium level was. So their selenium level was 1/4 of what the average level of selenium we find in the United States. And the healthy population was 2 to 3 times higher than the breast cancer patients. So certainly, if you are living in an area where you have excessive levels of heavy metals, they're going to burn up selenium and cause it, to be not utilized in antioxidants. It's going to be problematic. And again, the BRCA1 carriers are just a good example of women who have really are highly sensitive to environmental toxins such as heavy metals and so on and so forth, or maybe not having adequate levels of selenium. So there's a lot of information there. So again, for me, as I put this together, and this was in the book that I co-wrote with John Lee in 2002, how many millions of years ago was that, so 2002, 2022, we're 20 years ago, when we put this book together, this is what we were talking about, and that estrogens, estradiol, estrone are converted into both 2-hydroxy and 4-hydroxy estrogens. And the 4-hydroxy estrogens, we absolutely know those are the bad ones. But a 4-hydroxy estrogen has got to be converted into a quinone. And what we know is that peroxylipids, transhydrogenated fats, and heavy metals are going to speed that up. It's just anything that is a reactive oxygen species is going to speed up. It just strips electrons off of the catechol and makes them quinones. Those are extremely reactive molecules, and they bind to DNA. They form adducts. There's a ton of papers on this. They form adducts with DNA. They cause damage to DNA. So I go back to that, BRCA1. It's damage to the DNA caused by the oxidation products. But we certainly see elevated levels of the 4-hydroxy estrogens in women with active disease, and that's what we saw in the Chinese study that we did, where we looked at the levels of 4-hydroxy in urine. So that was published in 10 years ago. So higher levels of CYP1, but selenium and iodine right here are going to play a role to increase the levels of CYPA1, which is an enzyme that is more likely going to convert this to 2-hydroxy estrogens as opposed to 4-hydroxy estrogens. And the selenium and iodine are going to have impact on the heavy metals as well as the peroxylipids, because we're talking about selenium antioxidant, selenium enzymes that are effectively removing reactive oxygen species from the body. So selenium and iodine really play an important role. You can go to the literature and look. You can find books on iodine deficiency and increased risk of breast cancer, selenium and breast cancer. Just lots of literature out there on that. So this was my rendition of what I think is happening with regard to the oxidation of the the hormones. So again, estradiol, DMBA. I started my postdoc work because when I did my doctoral dissertation, I was working with dimethylbenzanthracene. We knew that if you give rats, young rats, not older rats, young rats, dimethylbenzanthracene, they develop breast cancer. They develop mammary cancers. They don't develop any other kind of cancers. They develop mammary cancers. And so that got me interested. And I went to the people that I began my postdoc with back in the 70s. And we were looking at estradiol. And then I got more and more involved in that. And I met Ercole Cavalieri and other people who were doing the work on catechol estrogen. So I definitely, once I started ZRT Laboratory, I said, okay, now I need to buy a mass spec and be able to start looking at catechol estrogens. And then we did that work. And we've been able to develop the assays to look at the 2 versus the 4 catechol estrogens. And we definitely see that women with active disease usually have much higher levels of the 4-hydroxyestrogens. That's another story. I'd be glad to lecture on that subject because I've done extensive work with that. So conclusion, selenium at appropriate levels is essential for optimal thyroid function and health. Remember, optimal levels. Paracelsus, he said that the dose is in the toxin is in the dose, something like that. You can have something that is really beneficial. Selenium is very beneficial at the right concentration, as is iodine, but not at excessive levels. People believe or seem to think that if something works well at one concentration that is physiologic, that they can bring it to pharmacologic levels and it's going to work better. It doesn't work that way. Mother Nature didn't make it that way. So optimal levels, remember those polo horses that all died because they got 100 times more selenium than they were supposed to get. Toxic metals like arsenic and mercury can lower the bioavailability of selenium and lead to adverse health consequences. Not going to kill you right off, but it's going to lead to more reactive oxygen species and more advanced aging. That's a problem. It's not something you notice on a day-to-day basis. There are tests you can do to see where you're at. We developed those tests both in dried urine as well as dried blood spot. We compared it ad nauseum to liquid urine and also whole blood, and we get same numbers. Our ranges are the same as National Institute of Environmental Health or NHANES or CDC. We're right on the mark with regard to expected levels. So we certainly can look at levels of iodine, selenium, arsenic, mercury to help identify deficiencies and excesses of these beneficial and toxic elements. Provide a rationale approach to lower. You, as a healthcare practitioner, can provide rational approaches to lower the levels of arsenic, mercury, increase iodine, and selenium, so that the levels are optimized. You don't need to give selenium to someone who lives in an area where selenium is sufficient. You do if they're living in an area. In the area that we live in, the Northwest, selenium is deficient. And I first heard about selenium because I have a farm, horses, and the vet came out and said, well, we're going to have to give the horse a shot of selenium because, they get a muscle disease. I thought, oh my gosh, I'm measuring selenium. I'm working on selenium. And he said, yeah, selenium is really deficient in the soil. So the cattle, they get locked up with a disease of their muscles. So selenium in the area that I live is important. So maybe not in other areas of the world where selenium is adequate in food. Like I said, in the plain regions of southern Canada, there's a lot of selenium there, and so the lentils they grow there has a ton of selenium in it. So eat lentil soup, you're gonna get all the selenium you need. So take-home messages or iodine should always be well balanced with selenium, always. So you really need to make sure, people measure iodine and that's all they measure. You really should be measuring selenium with it to make sure that they're well balanced. If selenium is low, based on testing, supplement to optimize thyroid efficiency. So you're going to be optimizing thyroid deiodenases. So you're going to be making, your cells are going to be able to make more T3 from T4. You're going to have better antioxidant capacity, and health benefits are going to be lower cancer and cardiovascular disease. I mean, those are the two main killers, aren't they? Don't use excessive amounts of the beneficial elements, iodine or selenium, as these can also be toxic at high dose. There was a fad to use higher doses of iodine, and they were realizing that pregnant women using excessive amounts of iodine, their babies were being born with goiters, because that's what happens if you, in some people, not everybody, but in some people, it can cause a shutdown of the formation of thyroid, and babies, like adults, need a good amount of thyroid hormone. If your arsenic or mercury are elevated based on testing, identify and eliminate their source. Could be your water. If you're drinking water from a well, you could have arsenic. It could be mercury from dental amalgams. Look at those studies they did, looking at kids. Consider a healthy lifestyle to slowly help rid a lifetime accumulation of heavy metals. And that's it for today. And I'm more than happy to answer any kind of questions that people may have. And you're more than welcome to write me. And if you have any detailed questions, dzava@zrtlab.com. It's pretty simple. You can look me up on, just put my name into Google and I come up all over the place.
Great.
So that's where we're at with regard to thyroid and iodine and selenium and heavy metals. And I thank you all for attending.
Thank you very much, Dr. Zava. That was an impressive, presentation. People have been praising you in the chat, I can see. So, and I've been following some of the questions, well, all the questions actually. And then, of course, we do have time for a few questions. And I think what kept coming up, I don't think anyone is in doubt here. Test, don't guess, kind of. But if there are low levels of iodine and selenium, a question comes up: is it best to have it as a food source compared to a supplement? If it's a supplement, what type do you recommend?
Well, I really don't recommend. I mean, there's so many supplements out there. Yeast is a good source of selenium. Iodine, as mostly, you can eat certain kinds of seaweed that have higher levels, but you might get some toxic elements in that too. But you can use iodine drops. You can use all different sorts of things. I use all of that stuff, all the way from Lugol's to, but small amounts, to taking iodine tablets that may have, I think there's one that's got 6 milligrams or something, or I might break it into quarters and take less than that. I don't take it every day. My iodine stays pretty good level, but there's just all sorts of sources. Selenium, mostly you can get that as a supplement, so you want to, you don't want to take too much of it, as I said. But what's sold as supplements is a good source of selenium.
Okay. There was also one asking, because now you were discussing a lot in regards to breast cancer. That's of course mainly women, but there was also like a question of like iodine and selenium in regards to sperm quality. Do you have a comment to that?
Well, certainly, there are books out there written about, again, just Google it, or whatever source you use. If you just type in iodine and breast cancer, you're going to get a lot of references. There's a lot of references on that, or selenium and breast cancer. That's, or the references that I put in here. It's a quick story. You're going to be reading for weeks.
It was more to sperm quality. If you had, but if that's not your expertise, you can just say no.
Well, I spent 2 years working in an IVF laboratory, so certainly dealt a lot with semen and sperm, and some were good swimmers and some were not. And I always wondered, okay, what's going on? Selenium is important as an antioxidant. Anything that is working as an antioxidant. I think it's for fertility. I think it's really important that you make sure that your iodine and your selenium are in good shape. It's just that's just a piece of that along with looking at your thyroid hormones, because in the very beginning I showed it's problematic if you don't have adequate levels. I think most parts of the world are not that deficient in iodine as they were 50 years ago, because the goiter belt was up around Michigan. It just doesn't exist anymore, because they supplement with iodine. And I know in China in the 90s, they started supplementing everybody with adequate amounts of iodine, but they didn't pay any attention to selenium. So now they have problems, of which I co-author on a paper, where iodine can cause some problems in individuals because it's there in the absence of selenium. I mean, you think about the thyroid gland is a factory. It's a hot factory. Thyroid peroxidase is taking iodine, it's ionizing it along with hydrogen peroxide. So it's a hot process. It's generating a lot of free radicals, but those free radicals are soaked up by the selenium containing enzymes that are present, extremely high concentration in the thyroid gland. So it prevents the iodine from damaging. That goes on all over the body. So selenium and iodine are important. They're, they work in synergy with one another, and they have to be balanced. And so if you have excessive heavy metals, you saw those diagrams, arsenic, mercury, lead, cadmium. Cadmium is associated with, high cadmium is associated with increased risk of breast cancer. So a lot of the heavy metals, what the CDC says are the top 4 problematic heavy metals are lead, mercury, arsenic, and cadmium. And there's good research out there on cadmium and increased risk of breast cancer. I didn't talk about cadmium. I was talking about the 2 heavy metals that are, reacting with selenium and pulling selenium out. So when you look at, you're looking at the essential elements, mainly the iodine and selenium. That's definitely not the only, but with regard to thyroid, zinc is too. But you've got to look at those relative to the toxic metals too. And again, those 4 are really important, because 2 of them, arsenic and mercury, are gonna bind to selenium and pull it out. It won't be used. It can't be incorporated into the enzymes that are involved in detoxification. And those enzymes are constantly being synthesized in the body, rapidly synthesized. You just have to keep producing those enzymes, because there's it's called enzymes suicide substrate, which is once they react with whatever it is they're reacting with, they don't last forever. The active catalytic site gets clogged up and it doesn't work anymore. So then they're eliminated, but you have to make new ones. So constantly making new levels of the antioxidants, as well as we know glutathione certainly is one of those. So,
and then a bright person is asking about bromine and fluoride and chlorine that goes in and blocks the receptors so that we get a deficiency and compete, but you're measuring that in some of your tests as well, in the urine test.
We look at bromine. We don't look at the fluoride or chlorine. Those are smaller molecules that are more difficult, by ICP mass spec to measure. George Gilson once told me, he said, yeah, they go up the smokestack. They're more difficult to monitor. So
then we have
to look at bromine.
Yeah. Okay. So then we have, I think, the last question, because people need to enjoy the rest of the day and the evening and so on. But there is one that I think, because I get that question quite a lot, like if you have a person with Hashimoto's or elevated TPO antibodies, and then like supplementing with iodine, of course, test so that you have an idea of the dietary intake, but there's this fear of giving iodine to someone who has Hashimoto's. Could you please comment on this?
Well, if they're iodine deficient and you give them a physiologic amount of iodine on it, I haven't seen that there's controversy over that issue. I definitely, people should look into the use of measurement of selenium in those diagrams that I showed. What it did was lower the amount of thyroid peroxidase, and it will suppress a thyroid storm. It's what you don't want. I mean, you can have an immune reaction to thyroid peroxidase. That's the enzyme that makes the thyroid hormone. So, and it's supposed to just be in the thyroid gland. So the immune system attacks the thyroid, and then it destroys the thyroid, and all this excessive thyroid is released into the bloodstream. So that's a thyroid storm. Selenium is working to increase the levels of antioxidant enzymes. So it has beneficial effects with regard to preventing a thyroid storm. It lowers, as you saw in those diagrams, it was lowering the level of the thyroid peroxidase enzymes, thyroid peroxidase antibody that's present in the bloodstream. So there's going to be less likely the problem of thyroid storm, because the thyroid storm is going to destroy the thyroid eventually. If it keeps happening, and you may get a viral infection and your immune system is kind of whacked out a bit, and that's when you may get more of a thyroid storm. I wouldn't be at all surprised if COVID being a viral infection isn't going to create more problems with that. Autoimmune problems, I think we know that that's associated with increased autoimmune problems. So I wouldn't be at all surprised. So important that you keep your elements at a happy level. But again,
But that sentence, keep your elements at a happy level. I think
maybe we should stop with that. And
that's a nice sentence to stop with.
Smiley face.
Yeah, smiley face. And I can see there's been a lot of smiley faces in the chat box, and Thank you very much, Dr. Zava, for your presentation and your time. And all of you out there, we will share the recording with you, and we will share the slides as well, so you can dig further in. I really appreciate the way all the research was like from different countries, so it kind of like fitted in with like we are from everywhere, and so was your research. So it just shows how it's a worldwide challenge balancing selenium, iodine, and thyroid profile.
It is.
So yeah.
So thank you very much for inviting me, and I'd love to come back if you want me to lecture on estrogen metabolites or any of the other things that we do.
I will definitely get you back here if you're interested in that. Thank you very much. And to all of you out there, see you on mail or another webinar, or maybe in live soon. Take care, everyone. Enjoy the rest of your day, evening, wherever you are. Bye for now. And thank you, Dr. Zava, again.
Bye.
If you do not already have an account with us, please contact our team and we will assist you. If you found this webinar helpful, please like and subscribe to our channel.